Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 23rd November 2020. As all of you know, we are into the revision mode. We are revising chapter number 10, cell cycle and cell division. As all of us know that we understand that the sequence of events by which a cell duplicates its genome, synthesizes its cell contents, and eventually divides into two daughter cells is termed as cell cycle. And a typical eukaryotic somatic cell has two main stages. One is the long undividing interphase, it's called I phase, and the shorter nucleus dividing stage called M phase or mitotic phase. And this is followed by cytokinesis. That is, cyto means cell, kinesis means division of cell. Virtually we can say division of cytoplasm. Interphase is metabolically active stage and is divided into G1 phase, S phase and G2 phase as we discussed. And we have also described in brief about the important characteristics of G1 phase, S phase and G phase. And in between, not to forget, G0 phase. Going ahead, we said that mitosis occurs during formation of body cells. Therefore, it is called somatic cell division. As the daughter cells resemble their mother cell genetically, it is also called duplication division or equational division. Mitosis leads to production of diploid daughter cells with identical genetic component and helps to restore nucleocytoplasmic ratio. It is also replace old cells and helps to increase the number of cells with an organism. And we also said in our last session that mitosis completes its turn by undergoing four phases. First one we said prophase, second one is metaphase, and third one is anaphase, which we need to discuss, and fourth one is the telophase. So basically anaphase, in anaphase, first and foremost thing is you can see the nuclear membrane disappeared. There is no nuclear membrane as such. And then cytoplasmic spindles are formed. Astral rays are formed. You can see the centromeres with astral rays. And you can also see that splitting of each chromosome at centromere into two sister chromatids. So anaphase is characterized by the presence of sister chromatids. Look at this. There is a virtual split of the chromosomes into chromatids. The two chromatids start moving towards opposite poles. So we said north pole and south pole. Now here in metaphase we said all the chromosomes, here see chromosomes are lying in the center of the plate, right? Whereas by the time the cell reaches to anaphase, first and foremost thing is chromosomes will split into chromatids. And the sister chromatids start moving in opposite poles, north pole, south pole. So this is the characteristics of anaphase. The spindle fiber splits the centromere and the chromatids move to opposite directions of the cell. That's the important feature. So first nuclear membrane disappears, first and foremost. Then the spindle fiber, formation of spindle fiber continues, and the spindle fiber splits the centromere of the chromosome and the chromatids, and the splitting of chromosome results in the formation of two sister chromatids, and the two sister chromatids start moving north direction and south direction. These are the characteristics, and we can see, most important thing is we can see the movement of chromatids into opposite poles. That's important. And entering into the final one, telophase is the last stage of mitosis, which involves chromosomes reach at opposite poles and lose its identity as a discrete unit. nuclear membrane reappears around the chromosome clusters. Nucleolus, Golgi complex, endoplasmic reticulum reappear. So what is important is, once the cell reaches the telophase, the chromosomes loosen on each side and the nuclear membrane begins to form around the chromatin. So strands of DNA will be formed. Now here, what we see when we observe a cell under telophase under the microscope is, first and foremost we can see the chromosomes have, in Anaphase we said chromatids will be movement of chromatids. Once they reach the poles, each chromatid duplicates. As a result, you get a chromosome. So you get one set of chromosomes on north pole and another set of chromosomes in south pole. So you got two sets of chromosomes, and then there will be duplication of these chromosomes. As I said, as a result of duplication of chromosomes, there will be retention of what we say 2n, diploidicity. So diploid number of chromosomes are retained. Because the cell retains diploidicity, the identity of the species will continue. The species will not lose its identity thanks to this telophase. And the astral bodies slowly will disappear. Cytoplasmic fibers also will disappear. And you can see there is a constriction. There is a constriction in the cytoplasmic as well as the cell membrane wall. So because of the constriction, slowly the constriction moves towards the center and virtually the cell splits into two parts. So when the cell splits into two parts, we can see two nuclei, one in the north side direction, another in the south pole direction. So the presence of two cells with the separate nucleus is the main characteristics of telophase. And once that happens, what happens? The nucleolus reappears because the centromeres will go off. Then Golgi complex will come back. Endoplasmic reticulum will appear. That means slowly all the cell organelles will restart appearing. And once this is done, cytokinesis is to take place. And cytokinesis is the division of cytoplasm of a cell. After karyokinesis, karyon means nucleus, kinesis means breaking of nucleus. So once the nucleus is broken, and this will be followed by cytokinesis. The karyokinesis, division of chromosome into two daughter cells, as I said, daughter cell 1, daughter cell 2. In animal cells, appearance of furrows in plasma membrane that deepens gradually and joins to divide cytoplasm into two parts. As you are saying, that furrow will be there. In plant cells, wall formation starts at the center and grows towards the outer side to meet lateral walls. The formation of cell wall begins with the formation of cell plate. So in case of plant cells, the cell wall starts from center and extends to sides. Whereas in case of animal cells, the furrow starts from one end and reaches the other end. That is the significance of this cytokinesis. So we must be thankful to cytokinesis. Because of that cytokinesis only, daughter cells are formed. And what is the significance of mitosis? What way mitosis is useful? Mitosis produces diploid daughter cells. This is the importance of it. Diploid daughter cells with identical genetic component. So the species, the cell identity is incompact. It helps in repair of cells. Whenever there is an injury for us, there is a depression, there is loss of skin, loss of muscle, loss of blood vessels. All these get repaired because of mitosis, thanks to them, and especially in the lining of gut and blood cells. And meristematic division in the apical and lateral cambium results in continuous growth of the plants. Thanks to continuous growth, thanks to mitosis, because of the presence of mitosis in plants grow throughout life. In our case, the mitosis will be helpful in repair, replacement of the cells, And of course, there is also one thing that we need for a certain age, until let's say a human being is 18 to 21 years, the person starts growing both vertically and horizontally thanks to the mitosis. But at the same time, one must remember that where there is a plus, there has to be minus. The mitosis is very good in all these aspects, but as I said, keep remembering, whenever there is a disturbance in the cytoplasmic and nucleoplasmic ratio, it results in uncontrollable mitosis. Uncontrollable mitosis leads to formation of tumors, and tumors further growth results in cancer. Hence, one must remember that uncontrollable mitosis leads to cancer formation. But how to stop it? What makes the nuclear or nucleoplasmic and cytoplasmic ratio get disturbed? That's a $1,000,000 question. Various factors are there that will disturb this ratio. Hence, well, basically, as a biology teacher, I would tell you that you need to have a good lifestyle. Any disease for that matter, irrespective of origin and source, the only main reason is our lifestyle, stress. The moment we take stress leads to varieties of diseases including cancer. Hence, lifestyle is important. Have a peaceful life, have a happy life. Take care.